Welcome to today's deep dive. We are incredibly excited to have you with us because today we are doing something a bit different. We are stepping into the roles of university immunology professors and senior instructional designers to deliver a really rigorous, custom-tailored masterclass for you.
Exactly. We're taking off the standard conversational hats and treating this as a comprehensive, long-form audio lecture on the incredibly complex world of autoimmunity. Our mission today is to completely map out this intricate, beautiful, and honestly, sometimes terrifying biological system.
Yeah, we want to know how the body distinguishes between over 100,000 of its own self-proteins and foreign invaders.
Ah.
And what exact mechanisms just completely break down when that vital distinction fails.
Right. And we are going to cover 100% of the concepts provided in our source material. We're not rudging, we're not summarizing superficially. We are diving into the mechanistic depth of every cell type, receptor, and signaling cascade.
And to give you an initial hook, just looking at the epidemiological data from our sources, one staggering fact jumps right off the page. Autoimmunity affects women at significantly higher rates than men. For some devastating conditions, we're looking at a 10-to-1 female-to-male ratio.
It's a striking demographic reality, and it's a thread we are going to pull throughout this entire lecture. Now, for our structural roadmap, we're going to follow a strict 5-module progression today.
Okay, lay it out for us.
Module 1 is tolerance mechanisms. Module 2 is etiology and pathogenesis. Module 3 covers type 2 autoimmunity. Module 4 is type 3 autoimmunity. And finally, Module 5 is type 5 autoimmunity and treatments.
Okay, but before we even get into the pathology, like the actual diseases in the later modules, I have to push back a bit. How is it mathematically possible for the immune system to just know 100,000 of our own proteins to begin with?
I mean, that is the foundational question, isn't it? And the answer introduces us directly to Module 1, which is tolerance mechanisms. To understand disease, we must first deeply understand the baseline of health, and that baseline is autotolerance.
Okay.
So autotolerance is divided into 2 distinct geographical phases. I want to do a comparative segment here right up front. We have central tolerance and peripheral tolerance.
The central tolerance is essentially boot camp, right? It's happening in the primary lymphoid organ.
Exactly. And peripheral tolerance is like the police force out in the body's tissues. Let's look at central tolerance first, specifically starting in the bone marrow with our B cells.
Okay, the lymphocytes that eventually produce antibodies, the mechanics here are just mind-blowing.
Yeah.
A precursor stem cell develops into an immature B cell, and it has to build a B cell receptor, or BCR.
Right, which is basically an immunoglobulin embedded in the membrane. But the bone marrow doesn't just hand the cell a pre-made receptor. The B cell randomly shuffles its own DNA, a process called VDJ recombination.
It's a biological random number generator.
Yes, and that randomness lets the immune system create millions of different receptors. But the glaring danger is that purely by chance, a B cell might generate a receptor that perfectly recognizes one of our own 100,000 self-proteins.
Which would be a lethal threat. So the bone marrow tests every single cell. It surrounds them with self-antigens. And if a B cell strongly recognizes a self-antigen, the bone marrow throws a red flag.
But, and this is crucial, it doesn't immediately kill the cell. The B cell gets a chance to fix its mistake through a salvage pathway called receptor editing.
I love this part. It literally reopens its genome.
Exactly. It targets the variable region, the V region, of its immunoglobulin. It swaps out genetic segments to alter the physical shape of the receptor. It's trying to mutate away from recognizing self.
So it edits the receptor, then tests itself again.
Right. And if the new receptor ignores self-antigens, it survives. But if editing fails and it remains highly reactive, the bone marrow initiates clonal deletion. The cell is forced to undergo apoptosis.
Apoptosis being programmed cell death. It just quietly dismantles itself from the inside out, so macrophages can clean it up without any inflammation.
Exactly. Now, what if the B cell receptor recognizes a self-antigen, but the bond is incredibly weak?
Our sources say it doesn't die, but it enters a state of anergy.
Yes, anergy. The cell is allowed into the peripheral blood, but its internal biochemical signaling pathways are permanently blocked. It is functionally silenced forever.
Okay, so that's the B cells. But T cells have an even more brutal education process in the thymus, right?
Oh, absolutely. T cells migrate from the bone marrow to the thymus as immature cells. When they arrive, they are classified as double negative.
Meaning they lack the CD4 and CD8 co-receptors. They're just blank slates.
Right. Then they mature, generate their T cell receptor, or TCR, and express both CD4 and CD8. They become double positive. This is when they face the first extreme test-positive selection.
And T cells are basically blind, right? They can't see free-floating proteins.
Exactly. They need proteins handed to them on a molecular serving tray. Those trays are our major histocompatibility complex molecules, the MHC molecules. Every person has a unique set.
So positive selection tests if the T cell can even interact with your specific MHC molecules. The thymic epithelial cells hold out MHC class I and class II.
Right. And if the T cell's receptor binds with a low functional affinity, it passes. It receives a survival signal. And right then, it makes a career choice.
If it interacts with MHC class I, it drops CD4 and becomes a CD8 positive cytotoxic T cell.
Exactly. A cell designed to kill infected cells. And if it interacts with MHC class II, it drops CD8 and becomes a CD4 positive helper T cell.
Which coordinates the immune response. And if it can't bind to any MHC molecule at all,
Apoptosis.
Yeah.
It dies by neglect. But this creates a massive paradox. We just positively selected T cells specifically because they bind to our own MHC molecules, displaying our own self-peptides.
right, we just deliberately selected for autoreactivity. That sounds like a recipe for disaster.
Which is why positive selection is immediately followed by negative selection in the medulla of the thymus. The medullary cells present thousands of self-peptides from all over the body.
Liver proteins, lung proteins, all loaded onto MHC molecules.
Exactly. And if the T cell's TCR binds to that self-peptide MHC complex with a high affinity, meaning it locks on tightly, that cell is incredibly dangerous. The fate for those cells is, again, apoptosis.
Usually. But there is a really vital alternative pathway here, right?
Yes. Sometimes a highly autoreactive CD4 positive T cell is bathed in a specific cytokine transforming growth factor beta, or TGF beta.
And that cytokine completely alters its destiny.
It does. TGF beta forces this dangerous cell to differentiate into a regulatory T cell or a TREG.
It reprograms the most dangerous cells to become the ultimate peacekeepers. That's amazing.
It is. These TREGs survive to patrol the body and suppress other immune cells. But even with this massive culling, like 95% of T cells die in the thymus, some autoreactive cells will inevitably escape.
Which brings us to peripheral tolerance out in the tissues. Our sources detail 4 step-by-step mechanisms here.
Yeah.
Ignorance, energy, apoptosis, and suppression. Let's start with ignorance.
Ignorance is simple. The immune system never attacks because it never sees the antigen. Certain tissues are immunologically privileged sites.
Like the eyes, the central nervous system, and the testes.
Right. They are shielded by dense physical barriers like the blood-brain barrier. The antigens are sequestered. The immune cells are functionally ignorant of them.
Okay, but what if a rogue T cell wanders into a normal tissue, like the kidney, and encounters a self-antigen? That brings us to energy, and this relies on a strict 2-step verification process.
Exactly. Signal 1 occurs when the TCR binds to an MHC peptide complex. But signal 1 alone does nothing. The T cell needs signal 2.
Signal 2 requires a molecule called B7 on the presenting cell binding to a receptor on the T cell called CD28.
Right. Only when you have signal 1 TCR to MHC and signal 2 B7 to CD28 does the T cell fully activate. But normal tissue cells don't express B7. Only professional antigen-presenting cells do.
It's exactly like a 2-key nuclear launch system. The T cell has the first key, which is the TCR-MHC interaction. But unless an official antigen-presenting cell turns the second key, the B7-CD28 interaction, the launch aborts and the cell is permanently deactivated.
That is the perfect analogy. When a T cell binds to a kidney cell, it gets signal 1, but no signal 2 because there's no B7. That lack of co-stimulation paralyzes the T cell. It enters profound energy.
It's permanently silenced. Okay, the third peripheral mechanism is apoptosis, but this involves Fas and Fas ligand, or Fas-L.
Yes. Activated cytotoxic T cells express a receptor called Fas. It's basically a self-destruct button. And privileged tissue cells, like in the eye, express the Fas ligand.
So if a rogue T cell gets into the eye, the tissue's Fas ligand binds to the T cell's Fas receptor.
Right, and that binding initiates a lethal cascade of casposes. The tissue commands the T cell to commit suicide.
Brilliant. And the fourth mechanism is suppression, which involves those TREGs we made in the thymus.
Exactly. TREGs patrol the tissues, and when they recognize self-antigens, they secrete potent immunosuppressive cytokines, primarily interleukin-10 or IL-10, and more TGF-beta.
Like a wet blanket on the immune system, they create a state of partial tolerance, which actually suppresses B cells too, right?
Yes, because B cells usually require T helper stimulation. If the T cells are suppressed, the B cells are stranded.
Okay, so to synthesize Module 1, tolerance is this multi-layered security system of central editing and deletion, backed up by peripheral ignorance, energy, apoptosis, and suppression.
Exactly, which perfectly sets us up to announce Module 2, etiology and pathogenesis. because we need to understand what happens when all those layers are breached. Autoimmunity is inherently multifactorial. It requires a perfect storm of genetics, hormones, and environmental triggers.
Let's unpack the genetic factors. The sources emphasize the human leukocyte antigen system, or HLA alleles.
Right. HLA genes dictate the exact physical shape of your MHC serving trays. And specific variations, or alleles, are shockingly associated with specific diseases.
For example, HLA-DR2. If you have that allele, your relative risk for multiple sclerosis increases by 5 times, and MS has that devastating 10-to-1 female-to-male ratio.
Then there's HLA-DR3, heavily associated with Graves' disease, myasthenia gravis, and systemic lupus erythematosus.
And HLA-DR4 increases rheumatoid arthritis risk by 4 times, with a 3-to-1 female predominance. We also have HLA-B27 linked to ankylosing spondylitis and HLA-DR5 associated with Hashimoto's thyroiditis.
But having the allele doesn't guarantee the disease. It just means your cells are highly efficient at presenting the specific self-peptides that can start the disease.
The gun is loaded. But we also have to talk about hormones. That overwhelming female predominance points directly at estrogen.
Yes, estrogen is a highly potent immunomodulator. It actively enhances immune responses, particularly stimulating B-cell activity and antibody production.
So females have a naturally higher baseline of humoral immunity. Great for fighting infections, but statistically much riskier for producing autoantibodies.
Exactly. So we have the genetics and the hormones. Now we need the environmental trigger. Our sources outline 4 mechanisms of tolerance loss. Number 1 is the release of sequestered antigens.
Which goes back to ignorance. Physical trauma can breach those barriers. A harrowing example is sympathetic ophthalmia.
Right. Severe physical trauma to one eye ruptures the blood-retina barrier, spilling hidden intraocular proteins into the bloodstream.
The immune system has never seen these proteins, so it mounts a massive attack. It generates Th1 cells, CD8 T cells, and antibodies.
And tragically, these weapons circulate and attack the perfectly healthy, uninjured eye, potentially causing total blindness.
Wow. Mechanism 2 is molecular mimicry. This is when an infection acts as a Trojan horse.
Yes. For example, Streptococcus pyogenes, which causes strep throat. The immune system makes IgG antibodies against the bacteria, but due to bacterial homologies with human proteins, those antibodies cross-react with normal human myocardium, the heart muscle.
Which causes rheumatic fever. The strep is gone, but the immune system attacks the heart. Mechanism 3 is inappropriate MHC IKLI expression.
Normally, only professional antigen-presenting cells express MHC class II, but under severe stress, regular tissue cells, like in the thyroid, can go rogue. They express MHC2 and present their own self-antigens directly to CD4 helper T cells,
bypassing peripheral checkpoints entirely. And finally, mechanism 4 is polyclonal B-cell activation.
Right. Certain viruses, famously the Epstein-Barr virus, or EBV, act as polyclonal activators. They forcefully cause a massive, nonspecific proliferation of B cells across the entire immune system.
It's explosive, and statistically, you inevitably activate and multiply some of those energetic autoreactive B cells, forcing them to produce autoantibodies.
So that synthesizes the etiology: bad genetic luck, hormones, and environmental sparks. This leads us directly into Module 3, Type 2 Autoimmunity.
Module 3, this is where we look at the specific weapons. In Type 2, the pathology is antibody-mediated, right?
Yes. Autoantibodies, specifically IgG and IgM, directly target antigens bound to cell surfaces or the extracellular matrix.
Let's detail the destructive Type 2 pathologies. Starting with autoimmune hemolytic anemia. Step by step, what is the cascade?
Okay. Autoantibodies bind to normal proteins on the surface of erythrocytes, our red blood cells. This dense clustering strongly activates the complement system.
And complement deposits fragments, like C3b, onto the red blood cell. Opsonization.
Exactly. As these coated red blood cells pass through the spleen, resident macrophages detect them. These macrophages have Fc receptors that specifically bind to the Fc portion, the tail end, of the IgG antibodies.
Once that Fc receptor locks on, it triggers phagocytosis. The macrophage physically engulfs and digests the red blood cell.
Yes, leading to profound anemia. And clinically, understanding this Fc receptor-mediated phagocytosis explains why a splenectomy is a viable treatment.
Because you're physically removing the primary site of red blood cell destruction. You're removing the slaughterhouse.
Precisely. We see a similar mechanism in neutropenia, but the antibodies target leukocytes, destroying the neutrophils and leaving the patient highly susceptible to infection.
Okay, those target circulating cells. But what about fixed tissue? like in Goodpasture syndrome.
In good pastures, IgG forms against type 5e collagen, which makes up the basement membranes in the kidneys and lungs.
So the antibodies rapidly accumulate along the kidney's filtration barrier, activating complement right there in the tissue.
Right. This draws in swarms of neutrophils. They try to eat the basement membrane, fail, and violently release their lysosomal enzymes, shredding the tissue. It leads to severe glomerulonephritis and renal failure.
Terrifying. But type 2 isn't always destructive. Sometimes it's receptor altering, like in Graves' disease.
Yes, Graves' disease is a bizarre mechanism. It completely compromises the thyroid gland. Normally, TSH from the pituitary binds to the TSH receptor on the thyroid, stimulating T3 and T4 production in a controlled negative feedback loop.
But in Graves's, driven by a Th2 response, IgG autoantibodies act as molecular mimics of TSH. They bind to the TSH receptor, but they act as agonists.
It is exactly like throwing a brick onto the gas pedal of a car. The antibodies constantly stimulate the gland without any negative feedback control.
So the thyroid pumps out toxic levels of T3 and T4. It results in unregulated hyperthyroidism, characterized by tachycardia, weight loss, and protruding eyes.
Now, contrast that with myasthenia gravis, where antibodies block and dismantle receptors. The target here is the neuromuscular junction.
Right. Normally, nerves release acetylcholine, which binds to acetylcholine receptors on the muscle to cause contraction.
But in myasthenia gravis, IgG autoantibodies bind directly to these receptors. First, they act as competitive antagonists. They physically block the acetylcholine from binding.
Like a biological earplug. But it gets worse because the antibodies also cross-link the receptors.
Yes. A single antibody binds two receptors, pulling them together. The muscle cell senses this abnormal cross-linking and induces endocytosis. It swallows the receptors and routes them to lysosomes for acid degradation.
So you lose the receptors entirely, causing progressive muscle weakness, drooping eyelids, double vision. Clinically, how do we anchor this?
We treat it from two angles. We use pyridostigmine, an acetylcholinesterase inhibitor. It blocks the enzyme that cleans up acetylcholine, allowing a massive flood of neurotransmitter to outcompete the blocking antibodies on the surviving receptors.
And then we use an immunosuppressant like azathioprine to stop the rogue B cells from making the antibodies in the first place.
Exactly. So that is type 2's precision targeting of cell-bound antigens, which contrasts perfectly as we transition into Module 4, type 3 autoimmunity.
Module 4. This is immune complex mediated. Let's do a side-by-side comparison. Type 2 uses antibodies attacking fixed antigens, like bricks in a wall. Type 3 uses antibodies attacking soluble antigens.
Right, freely floating proteins in the blood. When IgE antibodies bind to these soluble antigens, they create heavy, floating molecular clumps called immune complexes.
And the terrifying scope of this is best seen in systemic lupus erythematosus, or SLE.
The tolerance breakdown in SLE is unfathomable. The body produces IgG against ubiquitous intracellular components, surface proteins, cytoplasm, phospholipids, platelets, and crucially, double-stranded DNA itself.
So as normal cells die and release DNA fragments into the blood, the antibodies swarm them, forming these massive circulating immune complexes.
And based on hemodynamics, these heavy complexes mechanically wedge themselves into the delicate endothelium of blood vessels, the kidneys, and the joints.
And wherever they deposit, their concentrated FC regions spark massive complement activation via the classical pathway.
Which generates anaphytoxins like C3A and C5A. These sirens pull in massive swarms of neutrophils.
But the neutrophils can't eat the embedded complexes. It's frustrated phagocytosis. They vomit their lysosomal enzymes and reactor oxygen species all over the host's tissue.
The physiological fallout is massive chronic inflammation, vasculitis, severe glomerulonephritis, and severe arthritis, which is present in 90% of patients.
It's highly variable and has a distinct demographic skew, affecting 1 in 500 African or Asian women. It's just constant, exhausting immune stimulation.
That summarizes the antibody-driven destruction. Now let's introduce Module 5, Type IV autoimmunity and treatments.
Module 5. We are leaving antibodies behind and entering direct cell-to-cell combat. Type IV is entirely T-cell mediated.
Right, driven directly by autoreactive T lymphocytes. Let's look at Hashimoto's thyroiditis.
Okay, the mechanism here. Autoreactive CD4 Th1 cells and CD8 cytotoxic T cells specific for thyroid antigens, like thyroglobulin, infiltrate the gland.
The Th1 cells activate and secrete the potent pro-inflammatory cytokine interferon gamma, or IFN gamma.
Which acts like a megaphone. pulling in monocytes and hyperactivating them into furious tissue-destroying macrophages right inside the thyroid.
Meanwhile, the CD8 T cells are forcing the thyroid cells into apoptosis via phospho-cell or lethal granzymes. The thyroid is physically dismantled and replaced by scar tissue.
Let's do a comparative segment here. Graves' disease versus Hashimoto's. Both are organ-specific to the thyroid.
Yes, but Graves is type 2. It causes hyperthyroidism via receptor agonism, overstimulation. Hashimoto's is type 5e. It causes hyperthyroidism via structural tissue destruction.
Polar opposite clinical outcomes on the same organ. Now, type 1 diabetes mellitus is also a classic type 5e, right?
Absolutely. Autoreactive CD8 T cells selectively infiltrate the islets of Langerhans in the pancreas. This is called insulitis.
And they specifically target and induce apoptosis in the beta cells, which are the only cells that produce insulin.
Right, and they completely ignore the neighboring alpha and delta cells. It's surgical precision. The patient permanently loses insulin production.
We also have to detail rheumatoid arthritis here. What's the mechanism for joint destruction?
It's complex. A hallmark is rheumatoid factor. This is an IgM autoantibody whose specific target is the FCC portion of the patient's own normal human IgG.
So it's an antibody attacking an antibody, forming massive immune complexes inside the synovial fluid, triggering complement and inflammation.
Yes, but it also involves type 5e mechanisms. CD4 Th1 and Th17 cells infiltrate the joint and secrete cytokines that hyperactivate osteoclasts.
Osteoclasts, the bone-absorbing cells. So they literally eat away and destroy the structural bone and cartilage.
Exactly. Finally, for type 4, we must cover multiple sclerosis.
Right, linked to the HLA-DR2 allele. Autoreactive CD4 Th1 cells breach the blood-brain barrier.
They are specific for myelin basic protein. When they encounter the myelin sheath insulating the nerves, they activate, secreting massive amounts of IFN gamma,
which hyperactivates microglial cells and macrophages. These macrophages physically strip and destroy the myelin, leaving the nerve axons exposed and scarred.
This physical disruption of electrical signaling causes the profound neurological deficits in MS, from motor weakness to total paralysis.
Okay, which brings us to the clinical pharmacology. How do we fight back? Let's anchor the treatments we've mentioned, starting with cyclosporine.
Cyclosporine is brilliant. For T cells to multiply, they must transcribe the cytokine IL-2. IL-2 is their primary growth factor. Cyclosporine blocks an intracellular enzyme called calcineurin.
So it physically prevents the T cell from transcribing the IL-2 gene. It starves the T cells of their growth factor so they can't replicate.
Right. Then we have azathioprine, an antimetabolite. To divide, cells must synthesize new DNA. Azathioprine aggressively inhibits the synthesis of purines and pyrimidines.
So it completely halts the massive cellular proliferation needed for an immune response.
What about corticosteroids?
Broad-spectrum sledgehammers. They enter the cell nucleus and broadly inhibit the expression of various pro-inflammatory cytokine genes.
So they just dampen overall inflammation. And methotrexate.
Methotrexate is the gold standard for rheumatoid arthritis. It binds to and inhibits an enzyme called dihydrofolate reductase,
which is required for folic acid metabolism, which is needed for nucleotide synthesis. So again, severely impairing DNA synthesis for dividing immune cells.
Finally, we have the biologicals. These are engineered monoclonal antibodies.
Like the anti-TNF-alpha drugs, adalimumab, infliximab, and etanercept.
Exactly. TNF-alpha is a master pro-inflammatory cytokine. These biological drugs are engineered to seek out and bind specifically to TNF-alpha in the blood.
They physically neutralize it before it can reach its cellular receptors. Okay, I have to push back here. If these drugs are just turning off the communication lines, starving IL-2, or stopping DNA synthesis entirely, aren't we just trading autoimmunity for severe immunodeficiency? You've hit the exact clinical dilemma. Yes. These are blunt instruments. They stop the horrific tissue destruction, but they undeniably induce a state of iatrogenic immunodeficiency.
We are weakening the exact system designed to protect us from infections and cancer.
It is a high-stakes balancing act, which is why I want to leave you with a final provocative thought.
What's the future of immunology looking like?
Well, all current treatments are top-down suppressants. But what if the future isn't immunosuppression? What if it's artificially inducing peripheral tolerance?
Like pharmacologically synthesizing the fast ligand.
Exactly. Imagine attaching a synthetic fast cell specifically to the beta cells in a diabetic patient. Those cells could command attacking T cells to die, leaving the rest of the immune system perfectly intact.
Or extracting white blood cells and genetically engineering custom tregs.
Yes. Tregs engineered to recognize only myelin basic protein. infuse them back into an MS patient, and they travel to the brain, secrete TGF-beta, and tell the immune system to stand down on just that one protein.
Effectively curing the disease without turning the immune system off. That is incredible. Well, thank you for joining us for this rigorous masterclass deep dive. We've gone from central tolerance boot camp to the systemic storms of SLE to the cutting edge of biological pharmacology.
It is a staggering biological supercomputer. Keep questioning the mechanics and never lose your appreciation for the complexity of the biology keeping you alive.
Until the next deep dive, take care.